आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सॉल्यूशन पाओ। डाउनलोड नाउ।
In this question, we have to talk about the chemical change that proinsulin has to undergo to be able to act as mature insulin. Now we all know that insulin is a very important hormone in the body not only of human beings but also of many animals. And this hormone is secreted by the pancreas to keep the metabolism or the metabolic rate under endocrine control. So we see that in those people where insulin is not synthesized in the adequate amount, they suffer from diabetes because insulin is known to lower the level of blood sugar or blood glucose. But in the absence of insulin, we see that the blood sugar goes up and the person suffers from diabetes. So such people need regular administration of this hormone, which is nowadays being produced on a mass scale by biotechnological processes. So what we find in the human body is, if we look at the structure of the insulin hormone, it is produced as a prohormone. So we can say that it is produced in an inactive form. It is not completely functional when it is synthesized, because if we look at the structure of insulin, the mature insulin has two polypeptide chains, which we are going to call the A chain and the B chain. And in the functional insulin hormone, both of these chains are linked together by some disulfide bonds or disulfide bridges. But what happens here is that when the prohormone is formed, it contains an extra chain or an extra stretch called the C chain, which prevents it from being fully functional. So the main challenge that we faced while producing insulin in the laboratory by recombinant DNA procedures was how to remove this C chain and how to assemble our insulin hormone into mature form. But now this challenge has been met because an American firm called Eli Lilly has introduced the genes for producing the A chain and the B chain in separate E. coli plasmids, and both of these chains have now been produced separately, and after being synthesized separately, then they are joined by disulfide bridges. So we can totally skip the C chain and we can produce functional mature insulin, which can be administered to the diabetes patients. So what is the chemical change that proinsulin is going to undergo to become mature insulin? Proinsulin is an inactive form of insulin, which apart from the A and B polypeptide chains contains an extra stretch called the C chain, which does not allow it to be fully functional. And once this C chain is removed and the A and B chains are joined together by disulfide bridges, now we have the mature insulin.
क्लास 6 से 12 से लेके नीट, IIT, जेईई मेंस और एडवांस के लेवल तक 10 मिलियन से ज्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।